Hello everyone, my name is Jackson Blough and I was the SCI intern for the Wilbur Lab this summer. My project concerned natural killer cells and cancer immunity. A tumor is an abnormal growth or mass of cells that can expand and destroy tissue. If that growth is uncontrolled, the tumor can become malignant and cancerous. Our body's tumor immunity is accomplished by cytotoxic immune cells that search for and destroy cancer cells in the body. In response to something foreign being discovered in the body, the human body has two immune responses. The first of which is the innate immune response, which is nonspecific and targets all foreign cells and substances found in the body. In the innate immune response, natural killer cells, macrophages, and other immune cells seek out and destroy these foreign cells. The second immune response of the human body is called the adaptive immune response. This response is specific, meaning that it combats all cells marked with a specific antigen. B cells make antibodies, and T cells seek out and destroy cells marked with this specific antigen. This summer, our project focused on the innate immune response of tumors, and specifically natural killer cells. There are two classes of natural killer cells. The first of which is peripheral natural killer cells, which can be thought of as normal and functional immune cells. Peripheral natural killer cells are found in the blood and are cytotoxic, meaning that they can kill other cells. They typically express cell surface markers CD56 and CD16. The other class of natural killer cells was discovered within the last decade in the pregnancy microenvironment. These cells are called decidual natural killer cells and are angiogenic and promote cell and tissue growth, rather than being cytotoxic. This function makes sense, as pregnancy can be argued as similar to cancer, as both promote cell and tissue growth. Decidual natural killer cells can be identified by the CD56 cell surface marker, but typically do not express CD16. In kidney tumors, both classes of natural killer cells are found. While at first unexpected, this makes sense, as the presence of a tumor indicates that the cytotoxicity of peripheral natural killer cells is not functioning correctly. It can be suspected that something in the tumor microenvironment causes a phenotypic change of the peripheral natural killer cells into the decidual natural killer cells. These figures shown are flow cytometry data which indicates the presence or absence of cell surface markers CD56 and CD16. From this figure on the left, we can see that the natural killer cells in the blood are positive for both CD56 and CD16, indicating that they are peripheral. In the figure on the right, we can see that the natural killer cells found in tumors are significantly more negative for CD16, suggesting that they have a similar phenotype to decidual natural killer cells. In a previous experiment that cultured both cancer cells and natural killer cells together, we can observe that natural killer cells found in the blood are much more cytotoxic than natural killer cells harvested from kidney tumors. From this data, we can conclude that natural killers from kidney tumors cannot effectively kill cancer cells. In a summary of supporting data, we found that natural killer cells from kidney tumors are mostly CD56 positive and CD16 negative, and also have a poor ability to kill cancer cells. This would indicate that they are very similar phenotypically and functionally to decidual natural killer cells. Additionally, we found that something in a tumor microenvironment is being produced that converts healthy natural killer cells to these decidual-like natural killer cells and making them lose their cytotoxic function. In our research program, our goal is to determine what causes these natural killer cells found in tumors to become decidual-like, and also how to regain cytotoxicity in these natural killers so cancer cells can be killed. Our hypothesis is that the ability of natural killer cells to effectively kill cancer cells is inhibited by transforming growth factor beta, or TGF-beta, which is a pleiotropic factor released by tumor cells. TGF-beta has many functions across the body. To perform experiments with natural killer cells, we needed to harvest them from the blood of healthy and cancer-free volunteers. This was the primary procedure that I performed this summer. This process began with taking whole blood and separating it into its components through centrifugation. From here, we harvested the buffy coat, or lymphocyte layer, that contained natural killer cells and other immune cells. We then isolated natural killer cells from the rest of the lymphocytes via negative selection with dynabeads. Dynabeads utilize immunomagnetic selection, meaning that they contain small magnets conjugated to antibodies, which bind to the rest of the lymphocytes. So, we used a magnet to separate the lymphocytes from the natural killer cells, which allowed us to isolate them. Finally, after this procedure, we used flow cytometry to determine the composition of the lymphocyte layer and the purity of the natural killer cells we isolated. After isolating natural killer cells, we then performed a cytotoxicity assay to determine if these cells can kill cancer cells. Specifically, we cultured 2 cancer cell lines, the first of which is CACI2, a kidney cancer, and we also cultured K562, a type of leukemia. From here, we combined the natural killer cells we isolated with these cancer cell lines. The results displayed from the cytotoxicity assay confirm that the natural killer cells from whole blood can effectively kill cancer cells. We also performed a TGF-beta ELISA with the CACI2 cell line to confirm that the cancer cells do in fact produce TGF-beta. This procedure uses TGF-beta antibodies to bind to any TGF-beta produced by the cancer cells. The bound TGF-beta antibodies then bind to a secondary antibody, which can be visualized by spectrophotometry and determines concentration. The TGF-beta ELISA procedure revealed that the CACI2 cell line produces 4 nanograms per milliliter of TGF-beta per 1 million CACI2 cells. Another procedure we performed this summer was flow cytometry to determine if natural killer cells have TGF-beta receptors on them. As seen by the population marked in blue compared to the blanked population in gray, TGF-beta receptors are in fact present on natural killer cells. The final procedure we performed this summer was the same cytotoxicity assay that we performed before, but this time we added 2 nanograms per milliliter of TGF-beta to the natural killer cell cultures to evaluate differences in cytotoxicity between natural killer cells in the presence or the absence of TGF-beta. As seen from this data, TGF-beta was determined to significantly impair the cytotoxic function of natural killer cells. From our research this summer, the main conclusions that we found was that human natural killer cells express TGF-beta receptors, cancer cells produce TGF-beta, and that TGF-beta is a potent inhibitor of natural killer cell cytotoxicity. From here, I believe the next research questions to be answered include: what is the minimum exposure time required for the inhibitory effect of TGF-beta on natural killer cells rather than 48 hours? Can the inhibitory effect of TGF-beta be blocked by a small molecule inhibitor or TGF-beta specific antibody? How to stop this TGF-beta from its inhibitory effect? What gene expression changes are associated with the TGF-beta mediated inhibition of natural killer cell function? What is the mechanism? And are these results reproducible in vivo rather than just in cell culture? I'd like to take this time to thank all of the people on this slide for their involvement in this study. Specifically, I'd like to thank Dr. Wilbur, Chris Chambers, Hunter Jones, Joey Mayako, and Melissa Roberts for a great summer. I found this science fascinating and really enjoyed it. Thank you.